Good afternoon again. I'll be introducing our speaker, all the way from the FDA. And Dr. Yang is currently a policy lead in the Guidance and Policy Team, or GPT, under the Office of Clinical Pharmacology, part of CDER, Center for Drug Evaluation and Research at the US FDA. He graduated from the Department of Pharmaceutical Sciences of the State University of New York at Buffalo. He was mentored by Dr. Marilyn Morris. Currently, I noticed that you serve on a lot of different committees, which is very impressive. He's chair of Transporter Focus Group of the International Society of Studying Xenobiotics, or ISSX. He's also a committee member of the Regulatory Affairs at the same Society, International Society of Studying Xenobiotics. He's chair of the Membrane Transporter Committee at ASCPT, or American Society of Clinical Pharmacology and Therapeutics. He's also a committee member of PBPK community of ASCPT, and a member of the National Transporter Consortium Committee, as well as part of the ASCPT. Today, we're very glad that you're here to give us some regulatory science on transporters-mediated drug-drug interactions, and I know some of the labs here have done incredible research, like Dr. Giacomini and Sukwa, on transporters-mediated drug-drug interactions. So yeah, thank you for being here today, and let's get started.
Thank you very much, Silver. Good afternoon, folks. Thank you very much for spending time to come attend the seminar. It's my great pleasure coming here. You know, my background is biology in the undergraduate and the master study. So I did a lot of work in basic biomedical research. And the UCSF, everyone in this field knows this name because it's the top institution worldwide in many areas for biomedical research. So it's wonderful for me to come visit the campus. So my topic is, as Silver said, sharing with you on regulatory science on transporter-mediated drug-drug interactions. And here is just a typical disclaimer. This represents my personal view, not necessarily the official position of FDA. And here is an outline. First, I will share with you a short background, and then, talk about the transporter-mediated DDI from different perspectives, and with a summary. So this slide just to show you that clinical pharmacology is an integrated part of drug development and also regulatory review. So we answer a number of questions. And to access whether the drug is safe and effective. So clinical pharmacology will provide supportive evidence to show the efficacy of a drug. And also, what's the appropriate dosing regimen for the general population. And not only that, we also pay attention to subpopulations with different intrinsic or extrinsic factors. For example, drug-drug interaction is one of the factors that can affect the concentration of drugs and the pharmacology, the safety or efficacy of drug, so that we can ensure that we will provide appropriate dosing regimen to individual patients based on these factors. And as you know, this is an old slide, but as you can imagine, the trend is there that the aging population continues to increase in U.S. actually across the world, and the elderly population, they tend to use multiple medication, and when this happens, there could be drug-drug interaction, and the more drug you use, the higher likelihood of a drug interaction happening. So, back to quite a while back, Dr. Cassie Giacomini wrote a very nice review summarizing the examples where, drugs approved got withdrawn later on because of some safety concerns. And among this, several drugs I highlighted here, they are related to drug-drug interaction because, when they're concomitantly used with some other drug, this may significantly increase the concentration of the drug and hence increase the adverse event and sometimes lead to death. For example, this one, cerevastatin is one of the statins approved. It was found after approval that there are multiple reports of death. I think maybe 50, 60 deaths when those patients used gemfibrozil, a concomitant medication, to treat the indication like diabetes or something. So this is because gemfibrozil increased the AUC of cerebrastatin several folds. This is because gemfibrozil and the gemfibrozil glucuronide conjugate, they inhibit the CYP2C8 metabolism enzyme, and also it inhibits OETP1B, which transport cerebrastatin. So this highlights the importance to assess the DDI liability during drug development. So it's the good thing is that since then there are no such case happened. This is because we have done a great job by thoroughly evaluating the DDI liability of drug during drug development. But still, in some drug labelings, actually you can see here an example showing that there could be some drug interaction happening, and this will be managed and handled according to the labeling. So here I showed you several several DOAC, that's direct acting oral anticoagulants. Such drug gain more market share used by more and more patients compared to warfarin, because warfarin need therapeutic monitoring. So this DOAC, they don't need therapeutic monitoring, so they have an advantage than warfarin. Several of these are PGP substrates. So when such drugs are concomitantly used with other medications that are PGP inhibitors, their concentration may increase and lead to bleeding risk. And when they used with inducer of PGP, such as rifampin, their concentration will decrease, so that there could lead to more risk of a stroke. So the dose for these drugs you need to be adjusted, or sometimes may be avoided use. So because of the clinical significance of drug-drug interaction, FDA continuously look at this and issue several guidance. This traces back to 30 years ago, in around 1996, 1997. That's the initial, in vitro guidance, and then the in vivo guidance published. And in 2006, these two were combined. At that time, PGP was introduced into the guidance. And then by 2012, that's an update of FDA draft DDI guidance. Several transporters were added. This reflects the advance for science, because we know more and more transporters get involved in drug disposition. For example, OETP1B, OET, OCT. And by 2017, another two transporters were added, MATE1, MATE2K, because they often work along with OCT transporter-mediated renal elimination of a number of drugs. And in 2020, this guidance was finalized. So the goals of a DDI program during drug development has twofold. One is to look at the effect of other drug on the investigational drug, and on the other hand, we want to know whether the investigational drug will have an effect on other drugs. So the purpose is to first knowing that whether there could be drug-drug interaction happening, and if needed, this may be categorized with clinical studies to know the magnitude of DDI. And when you get that effect, you always need to put it in the context of dose or exposure to efficacy or safety relationship to understand the clinical significance of the interaction. So, like a statistically significant interaction doesn't necessarily mean that there will be clinical significant interaction. And for those clinical significant interactions in the drug product labeling, we will write a language to help patients and healthcare provider how to mitigate the risk. So about the timing of DDI evaluation, this usually start early because we want to screen for the DDI potential before give the drug to patients, so that this will determine the need and the timing of a clinical DDI evaluation. So the purpose, one, is to avoid give drug to patients, when there's a significant DDI which can affect the efficacy or safety of investigational drug. But on the other hand, we also don't want to exaggerate the risk because in that you will put an unnecessary limit on the concomitant medication, and this could affect the enrollment of patients into trial. Or, in the trial, because this drug is excluded, they won't reflect the real-world situation. So the DDI evaluation of a drug development, typically consists of the following parts. One is about, CYP enzymes for drug as substrates, as inhibitors, or as inducers, and similarly for transporters. And also, metabolites of drugs sometimes also carry the DDI liability. So what transporter to evaluate? And I copied these figures from a recently published review paper, with a group of experts for International Transporter Consortium, ITC. And from a regulatory perspective, we usually ask evaluation for the following transporters: PGP and BCRP. These are efflux transporter present in intestine and multiple other tissues. And OATP1B1, OATP1B3, they are the major hepatic uptake transporter, often transport ionic compounds. And the renal transporter OAT1 and OAT3, they are also often involved in the transport of ionic compounds. And OCT2, often work along with MATE1, MATE2K. That's more involved in cationic compounds transport. And besides this, routinely evaluated, there are some other transporter like OCT1, OATP2B1, and BSAP. This may be evaluated based on a case-by-case scenario. So first, let's look at evaluating investigational drug as a substrate of transporters with in vitro studies. So for PGP and BCRP, these are often conducted for orally administered drugs. The reason is that PGP and BCRP, they may limit the absorption of drugs. And in addition, for non-orally administered drug, if we know that this drug may undergo limited metabolism and biliary excretion or renal excretion is the major elimination pathway of such drugs, then we may also want to know whether they are substrate of PGP and BCRP, because this transporter is responsible for biliary excretion and renal active secretion of drugs. And in addition, if the drug target is in the brain, sometimes you also want to know if a drug is a substrate of these transporters, because they may limit the penetration of a drug into brain. But if it's a CNS drug, we hope that the drug will have a good distribution into brain, the target tissue. For OATP1B1, 1B3, because they are hepatic transporters, so if a drug, is undergoes significant metabolism or significant biliary excretion, we often want to know if they are substrates of OATP1B1, 1B3, because liver is the major elimination organ. For renal transporter, the thing is that drug may undergo glomerular filtration, which is a physical process not involving transporter. And besides that, they may undergo, active secretion process mediated by the uptake transporter OAT1, OAT3, or OCT2, and also the efflux transporter of MATE1 or MATE2K. So the focus is more on drugs known with a significant renal active secretion, which you can roughly calculate the contribution of that to the total elimination of a drug by use this formula. But just keep in mind that this equation actually has an assumption, assume that the drug does not have a real absorption, or say, the passive real absorption is, same as the active, so that it's cancelled out. For once we know that a drug is a substrate of a transporter from in vitro studies, then the next question is what to do next. Do I need to conduct clinical DDI studies? There are several factors to consider when you want to make that decision. One is the importance of transporter in the PK of the drug. For example, what's the passive permeability of a drug? In general, if a drug has a high passive permeability, the contribution of a transporter in the overall transport of a drug across the cell membrane tend to be limited. And the administration route, for example, if the drug is given by oral, non-oral route, some of the transporter in intestine, may not play a significant role. Also, the in vivo absorption and the elimination, sometimes you can get an idea from a mass balance study. And also, what's the clinical relevance of the PK change? So this, will be safety margin of the drug and the dose or concentration of a drug to its efficacy or relationship or safety relationship, so that we have a rough idea about, this much change of concentration increase or decrease, whether it could lead to significant clinical consequence. And also the likelihood of the drug to be used with concomitant medication that are modulator of a transporter as an inhibitor or inducer. So now let's switch the gear to talk about investigational drug as an inhibitor of a transporter. This usually starts also from an in vitro experiment to look at whether investigational drug can inhibit the following transporters. So once we know that if a drug inhibits the transporter, we measure, let's say, the IC50, and then we will use some simple equation, which I will describe in the next slide, compare that to the in vivo concentration can be achieved under the therapeutic dosing regimen to see how likely this in vitro finding will translate into in vivo. If we cannot rule out the DDI interaction, we have the several choices. One is go directly to conduct a clinical DDI study, which is a costly and also take time. But besides that, we also have some other tool. We can perform modeling, either mechanics statistical modeling or PPK modeling, to make a further assessment. And based on that, we can decide whether there's a need to conduct a clinical DDI study. So this is a snapshot summarizing the simple criteria from the different regional regulatory guidances. So you can see that majority of this actually are same or quite similar. And one thing to note is that these are based on empirical analysis. So this cutoff value can change when more data become available. So companies can propose an alternative cutoff. And one thing also to note is that this is used as the initial assessment. So this criteria tend to be conservative. And this, used to rule out the DDI potential if the ratio is below the cutoff value. But if the ratio is above the cutoff value, that does not necessarily mean there will be clinical significant interaction in humans. So, this is how such analysis were performed using this as an example for OETP1B. So basically, we collected a data set having two components. One is an in vitro IC50 of compounds on OETP1B, and the other set is the clinical DDI study conducted with this compound and drug substrates of OETP1B. So we say that, if this clinical DDI study show that the AUC of the substrate increase more than by 25% or more than that, we will consider that study as a positive study. And we will, compare the ratio shown in the previous slide and to this clinical study results. We can put those into four categories. One is, true positive. That is, the ratio is above the threshold, and the DDI study results is, above 25%. Or this can be, false negative, meaning that the ratio is below the threshold, but the clinical DDI study showed that there's still AUC increase more than 25%. And similarly, we will have a false positive and a true negative. So the purpose is trying to find the criteria which can minimize the false negative rate. But as you can see, by doing that, sometimes we are at the cost of substantial false positive, meaning that you thought that the drug will inhibit this transporter in vivo, and you continue to conduct a clinical DDI study, but it turned out to be a negative study. There's no significant change of the substrate AUC. So, this will add the burden to the drug development. So we also want to control the false positive. And here is the utility of endogenous biomarker. This is a really exciting area in the past several years with very rapid advancement in science and also incorporated into drug development. Because this endogenous biomarker can provide additional clinical data to help us decide whether the finding from in vitro is really can translate into in vivo. And also it can also help with modeling. The PPG modeling, is a powerful tool, but somehow people found that the in vitro determined IC50 underestimate the clinical DDI effect. So for PPG modeling, you have to do some adjustment for that IC50. You have to reduce the IC50 by certain fold. The problem is that that's not a consistent value across the drug. It's depending on individual compound. So it's hard to know prospectively how much adjustment is needed for an investigational drug. So endogenous marker provide additional useful information to also help better categorize the in vivo IC50 of an inhibitor or transporter, so that it can improve the prediction from PPK modeling. So in the next several slides, I will share you some of the recent research finding and case example to illustrate the utility of endogenous biomarker. This is a very nice paper, in a special issue published on Clinical Pharmacology Therapeutics a few years ago. It summarized the typical drug substrates and also endogenous biomarker for different transporters. So if you are interested, I highly encourage you to look at this paper. And among this endogenous biomarker, copper-porphyrin 1, CP1, is the one with the most amount of data, and it looks the most promising one. So on the left panel, it shows that prototype inhibitor rifampin can have this dose-dependent effect on copper-porphyrin 1. And not only rifampin, another well-known inhibitor of OATP1B, cyclosporine, also can increase the level of copper-porphyrin 1. And on the right-hand side, you can see that the pharmacogenetic studies also show that copper-porphyrin 1 concentration will increase as what we expected, in the subjects having the mutant allele of OATP1B. They will have a higher level of copper-porphyrin 1 than the wild-type carrier. Actually, this nice work is from Suk and Kaisi. Last year, the IQ working group, that innovation, the quality consortium, that's a pharmaceutical company consortium. They have a working group on OATP1B endogenous biomarker. They did a nice analysis by pull the data from a publication and also some data from different companies. So this shows that there's overall a great correlation between the AUC increase of a drug substance of OATP1B versus the level of a copper-porphyrin 1, CP1, Cmax, or AUC. So here is a case example, fenebrutinib from Genentech. It was developed for multiple indications. In vitro, it was found as an inhibitor of BCRP and OATP1B. They measured the IC50, and then we can calculate the ratio as use the equation described earlier. And it found that the ratio exceeded the threshold, so that we cannot readily rule out the clinical DDI potential. And here are the clinical DDI study results. On the left-hand side, it shows that it increased. So I apologize. Somehow the left figure is not correct. It's supposed to be the figure for rosuvastatin. It increased rosuvastatin AUC, I think, by fourfold. Rosuvastatin, we know that it's a dual substrate of OATP1B and BCRP. So this effect could come from inhibition of OATP1B, but it can also come from BCRP. The good thing is that they also incorporated measurement of copper-porphyrin 1 and copper-porphyrin 3, and showing that those endogenous biomarkers were not affected by fenebrutinib. And this actually has some, additional supportive from other clinical DDI study. Because fenebrutinib is a CYP3A inhibitor, they did a study, look at what's the effect of that on midazolam, a probe substrate of CYP3A. They found that midazolam AUC was about, doubled. And they also did a study with simvastatin, and they found that simvastatin AUC was increased, a bit more than midazolam. And this is actually in line with our expectation on CYP3A effect. Simvastatin is a very sensitive substrate. It's even more sensitive than midazolam. There are multiple literature studies showing that simvastatin AUC was increased more than midazolam in the presence of CYP3A inhibitors. And then, we have this study with rosuvastatin. As you can see, Cmax was increased more than AUC, sort of also implying that the interaction may happen more on the intestinal part. And in combined with the biomarker results, so we can confidently say that this effect is primarily coming from BCRP inhibition, and it doesn't affect OATP1B significantly, because if it inhibits OATP1B potently, then we expect that the effect of simvastatin would be even more, because simvastatin is also substrate of OATP1B. So there's a conceptual flow chart how to evaluate DDI risk in a publication by my colleague and myself recently. So here we actually add one layer of the evaluation compared to the previous flow chart I show you. That is after the in vitro experiment, after the initial assessment using those simple equation calculated the ratio. If the ratio is above the threshold, we can look at the effect on this biomarker level. And if there's no significant change of those biomarker, then it's very likely that the in vitro finding does not translate into in vivo. But if there's a significant increase of the biomarker level, then that means that there's a possibility, it's more likely you will see that in vivo, so that it's warranted to conduct a further clinical DDI study. So the question, as you can see here, the key question is what's the change of the biomarker level, right? Meaning that there will be, change in their significant inhibition in vivo of a transporter. So from that IQ group, based on the data they collected, they did some nice analysis. They found that the Cmax ratio of CP1, if that's about 1.25, meaning that there may be interaction, there may be significant inhibition of transporter by the investigational drug. And the AUC ratio could also be considered. So this is very helpful to implement into drug development. So here is another example. As I mentioned to you, this endogenous biomarker can not only be used to facilitate the qualitative assessment, meaning that there's no or yes interaction. It can also be used in combined with PPK modeling to enhance some quantitative prediction. This is a paper published by Pfizer recently. They incorporate measurement of biomarker in the single dose escalation study on the left side and the multiple dose escalation study on the right hand side. And then, they did a PPK modeling to see, how well they can capture the biomarker data change. This helps refine the key parameter, that is in vivo Ki or IC50 of the drug on OATP1B. So here it shows that they have a good prediction from PPK on, this atorvastatin PK data. And from here, they further to predict the effect on some other untested statins. So, this show you the scenario where, this endogenous biomarker can be used along with the PPK. So one thing I want to mention is that because rosuvastatin is a dual substrate of BCRP and OATP, but there are some drug that is OATP inhibitor, BCRP inhibitor. So when people conduct a clinical DDI study, rosuvastatin is the most often used substrate because it's a very popular used statins. So the clinical DDI study with rosuvastatin will have a clinical relevance. The caveat is that it's a dual substrate, and quite a few BCRP inhibitors, they are also OATP1B inhibitors. For example, here there are two oncology drugs approved recently. They increase rosuvastatin AUC a lot. And in the drug labeling, it said that the drug may inhibit BCRP, it may inhibit OATP1B, so that you need to do something about this. But the question is that this effect comes from OATP1B inhibition or BCRP inhibition, right? It's like a phenylbrutinib case I just showed you. Actually, the effect comes from BCRP. So that this labeling, will unnecessarily, raise a flag on OATP1B inhibition. So, one way is to incorporate OATP1B biomarker in the drug development program so that we can tease out where this effect comes from. On the other hand, if there are good biomarker for BCRP, that's also help us, to know what's the effect come from. And also for PGP and the BCRP, I want to mention, although the regional regulatory guidance from FDA, from European, from Japan, they use the same one, but there's actually some space, to improve. Because those criteria, they use a dose divided by 250 milliliter as a very rough estimate of inhibitor concentration for PGP or BCRP. The thing is that a lot of drugs, they are BCS class 2 or class 4, meaning that they have a low solubility. So the dose may not be immediately they may not be dissolved in 250 milliliter. So this is overestimate the DDI risk. But there's also a problem if you just use the aqueous solubility of a drug to replace that dose divided by 250 milliliter, because by doing that, you ignore the contribution from bile acid to help solubilize the drug, and also some excipients in formulation, or formulation features could also enhance the solubility. So far there has been a good resolution to that. And this is a potential utility of a biomarker. I know that Dr. Cassie Giacomini recently did some wonderful research with clinical studies to explore the utility of some BCRP biomarkers, and there's also effort from industry, like Dr. Hongshen from BMS, they explored riboflavin as a BCRP endogenous biomarker. So this is an area, definitely need more research, and hope that in the future we could identify some good biomarker similar to CP1 for OATP1B. That will greatly help, refine the prediction from in vitro data to in vivo. So now I will switch to the topic about drugs as inducer of transporter. This area, we have less understanding compared to inhibitors or substrates. The thing is that there are no good in vitro models for evaluating inducer of PGP. KCO2 is often used cell system, but the problem is that KCO2 has low expression of a nuclear receptor regulating PGP. And the LS180 is another cell line derived from colon cancer cells. It has expression of a nuclear receptor, but the thing is that it cannot form cell monolayers, so it cannot be used to do the monolayer transporter function studies. But nonetheless, there are clinical data showing that PGP can be induced by CYP3A inducers, because PGP is also regulated by PXR, a nuclear receptor that regulates CYP3A expression. It's just that PGP seems to be less inducible compared to CYP3A. So here I showed you the results of several well-known CYP3A inducers on several PGP substrates that undergo minimal metabolism. So the effect here, you can see that it's mainly come from PGP. The thing is that, majority of PGP substrates, they are also substrates of other enzymes and transporters. And other enzymes and transporters, they can also be modulated by inducers. It is not uncommon for inducer to modulate multiple genes. So overall, when there's multiple transporters, enzymes, including PGP, are induced, the overall effect sometimes can be still significant, as shown by some number in this slide. So you can look into the citation, the paper, for a full list. So, how to evaluate this if we don't have a good in vitro model? So here we have to leverage some of the clinical data. So first, let's think about investigational drug as a PGP substrate. So whether a clinical DDI study is needed with inducer, let's say strong inducer of CYP3A, this will depending on the understanding about the elimination mechanism, whether there's also other enzyme or transporter involved besides PGP, because if there's others involved, the overall effect could be significant. And also the dose or exposure efficacy relationship, if it has a wide margin, even the concentration is reduced by a certain extent, you will still keep largely the efficacy, then this may not be a big concern, and also the likelihood of concomitant use with inducer. And on the other hand, for investigational drug as an inducer, whether a study is needed to evaluate the effect on substrate of PGP in vivo, it depending on the magnitude of this drug as a CYP3A inducer, because like I said, PGP is often co-induced with CYP3A, but less inducible than CYP3A. So if a drug is a moderate or weak inducer of CYP3A, its effect on PGP would be even less. So this may not be a big concern. And of course, also the likelihood of concomitant use and also the dose exposure to efficacy relationship of a PGP substrate. Very quickly, this is show you that OETP1B substrate can also be exposure can also be reduced by some inducers. Here is showing that the pravastatin and the rosuvastatin, their concentration was reduced by rifampin in a dose-dependent manner. However, there are some more complexity here. There are some controversy data based on in vitro, animal, or clinical DDI study data, arguing against that OETP1B can really be induced by activator of a PXR nuclear receptor. So, more research is needed to understand what the mechanism of OETP1B induction. But anyway, there are quite a number of clinical DDI studies showing that CYP3A inducers, they can reduce the AUC of rosuvastatin by a certain extent. So, here, majority of it is rifampin, and there are some additional inducers. So, there's still some area need further research. So I will lastly talk about some other transporter besides the one routinely evaluated during drug development. For example, this OETP2B1 is present in the liver and in the intestine. In the liver, because there's also OETP1B1 and 1B3. So in general, it seems that those may play a bigger role. So the focus is more on intestinal part. One thing I want to mention, although in this slide, OETP2B1 is shown on the lumen of intestine, the apical membrane, there's still some, argument, whether it's where it's exactly located. There are some people thinking that it's present also on the basolateral membrane. So, and there are limited clinical studies to show, OETP2B1 involved drug interaction. Actually, majority of this come from food beverage interaction. Here it shows that some like apple juice, orange juice, or grapefruit juice, it can reduce the concentration of some drugs like say fexofenadine. So there has been a few example about a drug as a OETP, a clinical use drug as OETP2B1 inhibitor. And this is a second case example I want to share with you. This drug, tenapanel, was approved several years ago. It's a local acting drug approved to treat irritable bowel wine syndrome, IBC. So it affects certain transporter in the intestine, and the company conducted a clinical DDI study and found that this drug reduced the AUC and the Cmax of enalapril, and also the active moiety of enalapril. And initially this was thought, well, maybe this is because it inhibits peptide 1 and the peptide 2 transporters. But the thing is that the company also conducted another study to evaluate the effect of this drug on cefadroxil, peptide 1 and peptide 2 substrate. And in this study, we didn't see the effect. So what might be the reason for that? And we think about OETP2B1, it's because unlike pre-LIT, the active moiety is a substrate of OETP2B1. And tanaparinol, we know that it inhibits OETP1B1 and 1B3. So this trigger us send a recommendation information request to the company, and the company did further in vitro study, in vitro experiment, and they found that first of all, enalapril, the prodrug, is also a substrate of OETP2B1, and they also found that tanaparinol is an inhibitor of OETP2B1 with a low IC50. So, this information combined together, lead to us in the labeling of tanaparinol, we mentioned that it's an inhibitor of OETP2B1. So when it used with drug substrates of OETP2B1, you may need to pay attention that it may reduce the exposure of those drugs. And lastly, I want to mention that there's an effort developing globally harmonized drug interaction guidance. This is from ICH, International Council of Harmonization. The final guideline is going to be released very soon, this month or next month. But having said so, there are still a lot of work needed to be done. So as a summary here, that drug interaction is an integral part of a drug development program. It's important to characterize the effect to ensure that for individual patients, we will provide appropriate dosing regimen to ensure the safe and effective use of drugs. The criteria of those simple equations, they could be updated once more data become available. We will look at those periodically. But there are some limitation for that, and the recent advancement in endogenous biomarker actually, help a lot with refine those criteria and also, provide some additional support for PPK modeling. There are still a lot of work needed to be done. For example, for the in vitro experiment, we know that there's some work around this albumin-facilitated drug transport. And also for drug as an inhibitor, there are some discussions or data around whether pre-incubation is needed. And for the mechanism and the clinical relevance of a transporter, especially OETP1B1, 1B3, it worthwhile further research. And also, mechanical statin modeling and the PPK modeling for transporter-mediated DDI, there is still, less material compared to CYP-mediated DDI. And, there are complex DDI situations involving enzyme and transporter. For example, you all know that a lot of drugs, they are dual substrates of CYP3A and PGP. And when they used with CYP3A inhibitors, there are some nuances because some CYP3A inhibitors, they are PGP inhibitors, but some CYP3 inhibitors have a minimal or no effect on PGP. And also there are a couple of PGP inhibitors that do not inhibit CYP3A. This is a CYP3A PGP interplay. This phenomenon was observed by Dr. Les Bennett, and he published research on this a while back. But there are still more work needed to do when we consider how many clinical DDI studies are needed on this. Also, I didn't talk about the DDI liability of metabolites. There are much less data compared to parent drug, compared to data around the metabolite on CYP enzyme. So we need to accumulate more data. And also here, I only talk about the impact of a transporter-mediated DDI on plasma concentration of drug. But as you know, transporter can help, can affect the tissue concentration, not necessarily reflected by systemic concentration, and this could affect efficacy or safety. And finally, I want to thank some of my colleagues, and also thank you all for your attention.